Today's class, I'll be covering these topics. I'll be going through like what is microarray, the brief introduction. I'll talk about the history of microarray, types, different types of microarray, different platforms and different repositories. And being a mature technology, it has a lot of platforms, a lot of repositories, and there are lots of types. I will discuss about those things. And then briefly, I will go over how this microarray works in general. And then I'll talk about the design, explain of design. So how, if you plan to do the microdata analysis, then how you should design the experiment, what should be the sample size, and how to do the power calculation. Those things I'll briefly cover. And broadly, there are many types of, but I'll discuss about two types of microarray: DNA microarray, that is cDNA microarray, or and oligonucleotide microarray. And Epi matrix is the oligonucleotide microarray. So I'll be talking more in detail about Epi matrix gene chip. And then lastly, I'll talk about the methods, different methods of analyzing the microarray data. Okay, let's begin. So what is gene expression? We all know very well, right? Gene expression is a process in which the information in the gene converts into the different functional products. So functional products are proteins. And we know the central dogma of molecular biology, the functional region of DNA will be transcribed into RNA. RNA will be translated into protein, amino acids. Amino acids, they form different shapes, structures, and proteins act alone and together to give rise to different cellular functions, right? And what is gene expression profiling? It's a measurement of gene expression is gene expression profiling. We can measure the gene expression, right? Gene expression. And it is important because it will help us understand the, because gene expression varies at different time point, at different physiological conditions, at different experimental conditions, and it will help us understand how the cell or tissues are functioning at that moment, at that particular time, right? And maybe, you know, if you do gene expression profiling, you may be able to see some oncogenes expressed, overexpressed. You may see tumor suppressing genes overexpressed, or you may see the genes, those are involved in cell division process overexpressed. So you can determine what's going on in the cell by studying the gene expression profiling. And different approaches, there are different approaches to gene expression profiling, sequence-based and array-based approaches. This is like in the sequence-based, you can, like, you know, there is a express sequence tags methods, there's a says serial analysis of gene expression, but most common is the RNA-seq, and we'll be covering about the RNA-seq in the next class. Next is the array-based, and it is a microarray. It is developed mid 1990s, and it's a very mature technology in compared to the sequence-based. And other than that, there are many others, like, you know, you have been using the qPCR, right? The RT-qPCR method. These methods is used to measure the gene expression for a handful of genes, not for all entire genome-wide genes, just handful of genes. And it is widely used for those methods. qPCR is used for validating the RNA-seq gene expression or microarray gene expression profiling. Okay, so there are two different approaches. So today's topic is on the microarray development, which is developed in 1990s. So what is microarray? Microarray, you see that the gene chip and the layout of the microarray here in the figure. So what is microarray? Microarray is a multiplex lab on a chip. It is a lab on a chip. And according to the Wikipedia, it is defined as a two-dimensional array on a solid substrate. It could be a silicon wafer or a glass slides that assess large amounts of biological materials using high throughput screening, miniaturized, multiplexed, and parallel processing and detection methods. So when you say array, it is a sequential order of certain elements, right? So in a microarray, there is thousands of spots arranged in an orderly manner. And each spot contains millions of copies of oligos or DNA molecules that uniquely really corresponds to a gene. Like each spot corresponds to a particular gene. So those molecules, those oligos, are designed in such a way that they can capture the designed gene. Like they are the complementary sequences to capture the respective gene, right? Spots either may either be genomic DNA or short stretches of oligonucleotide strands that corresponds to a gene. And these spots, these oligos or these molecules are printed on a glass slides by a robot, or it could be synthesized by a process called photolithography. Photolithography is a Greek word, right? You may have heard about this word before too, many times, right? Photo meaning light, litho meaning stone, graph means sketch or graph, you know. So, so photolithography is a way that uses the light to insert or to print the material based on the template on a silicon wafer, and it is a very expensive but very accurate method. And most of them, the chips in current days are fabricated based on photolithography method. And in short, microarray is a portable device. It's a tool. It's a small and portable, very small, right, chip that can monitor a genome-wide gene expressions. And each chip is used to measure the gene for one sample. Okay, if you are planning to do gene expression for one sample, then you can use one chip. But if you are planning to do the differential gene expression for different groups, then you need to do the multiple experiment, you need to have a multiple microarray experiments done. So a set of microarray experiments allows to find genes that are differentially expressed between two kinds of sample. And if you have different, like, multiple microarray experiments done per sample, then you can run statistics. You can get a difference in expression between the two conditions. It could be like difference between lung cancer versus normal, or it could be knockout versus wild type, or it could be different treatment conditions, right? You can do the two-way comparison. You can do multiple ANOVA type comparisons. For that, you need to have a good sample size. And that's not all. That's not all. Okay, let's go briefly to the history. The history says that microarray was first published in 1982. And it is not, in fact, it is a microarray-based paper based on visual scoring method, more subjective. There is a paper, I've given a link to the paper, you can go through that. This paper usually used to measure the expression of sequencing clones, and it just use the mouse samples, different mouse tissue samples and tumor samples, and try to understand whether the gene is highly expressed or average or no expressed. And there was no statistics involved. It was just based on visual scoring, right? No image processing, no computational tools involved, nothing. So it was a very simple, primitive method in 1982. Then in 1997, the array structure was adopted by plating colonies grown in microliters plates onto filters, and quantification was done by computer-based scanning. So here, the two things improved. One was array structures was adopted, and other was computer scanning, based scanning was incorporated. Then real microarray began in 1995. So there were two companies at the time, Elizand and Affymetrix. You must have heard about both these companies. They are very popular companies nowadays. So they started their business. They started in 1995, and if Elizand started the glass slides based approach. So they started printing the complementary DNA sequences on using a robotics arm onto a glass slides, and Affymetrix used the photolithography onto the chip. So they used in situ chip method, based in chip method, to build the microarray. And first microarrays were not based on gene expression profiling. They were not to capture gene expression. They were captured. They were used to see the protein expressions, proteins levels. And in 1996, Affymetrix chip representing full human mitochondrial genome was launched. And main application was not, again, gene expression here, but detecting polymorphism. In 1997, the Affymetrix chip representing complete yeast genome was launched. So this is the brief history. Then now it's 2020, we are in the 21st century. So things have changed a lot. Like, let's go through different types of microarray. But when I begin in the before this, when I started, I talked about gene expression profiling, gene expression, right? But microarray is not limited to gene expression profiling. It is not only for gene expression profiling. Uh, but still, I talked about gene expression because most of the microarrays are designed to profile the gene expression. And their DNA microarray is very common. So is in DNA microarray into cDNA microarray, oligonucleotide microarrays. These are very common methods, common type of microarray for gene expression profiling. Besides that, there is a BAC microarrays, SNP microarrays, and there is MM chip. This is for surveillance of microarray population. There is a protein microarray or peptide chip. This is for tracking the interactions and activities of proteins and to determine their function. There is a peptide or microarray or chip. This is for detailed analysis and optimization of protein-protein interactions. There's a tissue microarray for multiplex histology analysis. There is a cellular or cell microarray. This is for multiplex interrogation of living cells in which can interact with varying materials such as antibodies, proteins, lipids. There's a chemical compound microarray. For this chemical compound microarray is used mainly for drug discovery research by pharmaceutical companies. So it's for searching proteins that bind to specific chemical compounds. And there is an antibody microarray for detecting antigens. There is a carbohydrate array. This is for evaluating of interactions between carbohydrates and protein cells or viruses. There is a phenotype microarray. This is for quantitative analysis of thousands of cellular phenotypes. There is a reverse phase protein microarray. This is the protein microarray used to measure the protein expression levels. And these are not all. This is just like some of the types, right? You may know of different other types. So one type is a virochip. Last year, I was one of the presenter about virochip. Virochip is a microarray that measures the different types of viruses. This is for viral profiling, viral expression profiling. So virochip. There are different other types of microarrays. And commercially available microarray platforms, there are a number of commercially available microarray platforms. Some are very good in doing business, some are not. Some are very popular, some are not, but there are different many other types of commercially available microarrays. So Epimetrics, there is a Agilent technology, there is Applied, there is Biosystems, CloneTech, Combi Matrix, Epindof, General Electric Life Sciences. This is a very big company. General Electric does multiple things. As Illumina, still it's a number one in the sequencing business, but not in the microarray. But it does has a microarray platforms. There's a Morgan. There is a Roche Nimble Genes. So among them, and there are many others which I missed. So Nimblezine, among them, I've used Nimblezine and I've used Epimetrics and Illumina's microarrays before, but many I've never used them. And if you look about Epimetrics, the Epimetrics is the number one in microarray business, but now there is no company called Epimetrics. The Epimetrics was procured by Thermo Fisher Scientific. You may have heard about Thermo Fisher Scientific, right? It's a very big company. Its market value is in billions dollars, in multi-billion dollars. And still, you see, you'll find the Epimetrics name appear in the website. But whenever you see the Epimetrics, then Thermo Fisher logo or their trademark will appear together. Okay. Now, there are different microarray data repositories. There's a number one is Gene Expression Omnibus of NCBI GEO. So if you are submitting a paper using microdata, then you may want to submit the microarray data to this NCBI GEO database. And then there is an Array Express, and there is a molecular signature database from our JESA, from Broad Institute. And this JESA is being widely used for pathway enrichment analysis. It's called gene set enrichment analysis. And OncoMine from University of Michigan. So this is also one of the repositories. There are many, many repositories, but this is just few of them I'm showing here, just to give you the sense like how things are. Okay, now moving further, how does microarray how does microarray works? First thing is you need to collect the tissue sample, right? You may be collecting the tissue sample from different experimental conditions or different time point or different uh, stages of developmental stages. So you collect the sample, then you isolate the RNA from the sample, you extract RNA, then you make the complementary DNA on that. Then you add a level, fluorescence level onto that sample. Once you add the fluorescence level, then some people add spikes in probes there. This spikes in is mainly to measure the how accurate the system is, how precise your measurements would be. So for the controls, you add spike in controls. Then once you make the hybridization cocktail, you inject that to the chip, and then you let it hybridize for a while, for a day. And then once it's dried, then you are ready to scan it. And once you use the scanner using the laser beam of two different wavelengths, then you will see the colors. You will see each spots will glow based on the gene expression. So some will glow brighter, some will go darker, based on if it's a two-colored microarray, then you will see two different colors. Then once you get the raw image file, then you do the background corrections, you do normalization, and then you are ready to call. You got the signal now. Based on the signal, intensity, how high the gene is expressed, how low the gene expressed. Based on the signal intensity, you can determine it. So this is the overall general workflow, and I've written those methods in here in the slide. So I'll just would go through briefly, okay? So first is RNA is extracted from cells or tissues. Second is extracted RNA molecules are reverse transcribed into complete DNA by using an enzyme reverse transcriptase. Then third is you complete DNA is coupled with different fluorescent dyes. So two array microarray, what you do is you add you have a one set of experiment. You add one type of dye, like say you label with red fluorescence CY5. Then on other sets of experiment, other sets like control samples, you add another dye like green fluorescence CY3. For example, a certain knockout condition sample may be labeled with red fluorescence dye, and the wild type may be you label with the green fluorescence dye. So you label the sample, and then two differently labeled cDNA samples are mixed in equal quantity with a solution containing agents that will allow it to be hybridized to DNA microarray probes. Then labeled cDNA solutions is added to the microarray and cell. So you once you make the hybridization cocktail, you add that to the microarray and then let it sell and stay. This microarray is then temperature controlled and rotated to ensure optimal binding between the labeled cDNA and probes. So you let it sit overnight to complete the binding. So we need to make sure that the hybridization is very smooth, well done. And next is microarray is dried and is ready to be scanned. And for scanning, a special machines uses a laser to excite the labeling dye with a detector records its brightness. And usually a laser beam is used. A laser beam of two wavelengths is used. That's one for the red fluorescence and other is for the green fluorescence. And based on, and it is used in each spot, and the red and fluorescence signals are recorded. Two fluorescence images from the scanners are then overlaid to create the composite image which indicates the relative expression level of each gene. So the final experimental stage is the image. From microarray experiment, you get the final raw data is an image file. It is not like the RNA-seq where you get the count, read counts. In a microarray, you get the image files. So in this picture, it's defined, it's clearly shown. So one sample is labeled with green dye, another is labeled with the red dye, and then you excite these each spots with a laser beam. Then you will get the different uh, colors here depending upon which sample is overexpressed. If one sample is overexpressed, then each spot, the spot will be green, and uh, and if it is red is overexpressed, then it will be red. And if both are equally expressed, then it will turn to yellow. And if none of them are expressed, then it will turn to be black. So let me go through this. The final experimental stages is an image of the microarray in which each spot that corresponds to a gene has an associated fluorescence value representing the relative expression level of that gene. Amount of fluorescence emitted upon excitation corresponds to the amount of bound nucleic acid. And here, if cDNA from condition one condition, say this condition first, for a particular gene was in a greater abundance than that from the condition B, which is a red dyed, one would appear the spot to be red. If it was the other way, the spot would be the green. And if both are expressed in the same level, then it would be yellow. And if none of them were expressed, then the spot would be blank, would be dark black. The final raw data in Epimatrix microarray are compiled by a cell file. So the final data is a raw data, image data. And if you are using Epimatrix microarray, then you get the data file in a cell format, cell file. But there are different types of microarray, have different image format. It's not necessary that you will have a cell format, like some may have TIFF format. And the main thing after you get the image file is to locate and quantitate the hybridization spots. You want to separate the true hybridization signals from the background noise. And what is background noise? The areas without probes also gives a signal, and that is known as a background noise, background signal. And background signal is not constant everywhere. It may be different in different parts of the array. And computer program should be well written so that they can accurately locate the boundaries of the spots and measure the intensities of the spot images after subtracting the background signal. So to get the signal, you have to subtract, you have to negate the background noise, background signals, then you get the accurate signal. Yeah, so final raw data values depends on the good experimental procedure, and that is crucial to produce consistent cell-wide gene expression profiles. And at various stages, error can rise and be amplified. Microarray can contain errors from the manufacturing processes from the very beginning, right? And there are different background noises and artifacts. This could be produced due to multiple factors like non-specific hybridization, meaning that there are spots where there will be hybridization occurring which are not supposed to be, right? There's a non-specific hybridization is unintended binding, which untargeted binding, which is considered as a noise. There's unevenness on the slide surfaces, and there is a presence of contamination such as dust on the microarray surface of the slides. And especially if you look at the hybridization spots, like the hybridization spots, like these spots, there should be a randomness. There should be no any specific geometric pattern there. But if you see any irregular shapes, any specific geometric patterns like circle or spiral, any geometric patterns, then you know that there is something wrong, the hybridizations. So look for geometric variation of hybridization spots resulting in some spots being of regular shapes. That gives you the idea that something is wrong. The background noise is high, there's artifacts, all those things. So we'll look at this later. Data transformation normalization. So before we do any statistical analysis, number one thing we need to do is data transformation normalization. Without transforming and normalizing data, we cannot make the correct inference. Data transformation is to bring the data more a normal distribution, and if you are using a normal distribution, then you can use the parametric tests right? So usually in the microarray, you do the log-based, logarithmic base of base 2 transformation is more common. And the main reason of log 2 base transformation is to get the uniform distribution of the data. And also if you do log transformation, the up and down regulated genes are more symmetric. You'll see more symmetric in the up and down regulated genes expressions. Data normalization is important to correct the bias owing to variation in microarray data collection. And usually in microarray we use quantile normalization. It's very common. One of the assumptions of this type of experiment is that the cell produces consistent gene expression, so different expression can be identified. And obviously, we are doing this kind of experiment to understand what genes are differentially expressed, right? So, and this is from this type of experiment we can do differential expression analysis. Each microarray is designed to capture gene expression per sample, right? But if you want to go for a difference in expression analysis, then you have to have a multiple experiments done, multiple micro experiments done. And that multiple microarray experience will help you determine the variability in the data. And in statistics, variability is very important. And in the replicate data sets, you can use statistical tests like t-test or ANOVA. T-test to compare two groups, ANOVA is to compare multiple groups. And usually, to consider differential expressions, people have different criteria, selection criteria. Usually, in a published paper, you see that the selection criteria to call differential expressions are like fold change greater than 2 or less than -2, and p-value less than 0.05. And some people also use q-value less than 0.05. Less than 0.05, not 0.5. I made a mistake here. It's p-value less than 0.05. A q-value less than 0.05. The q-value is a FDR adjusted p-value is called q-value. Okay, many get confused. What is q-value here? So q-value is FDR adjusted p-value is q-value. So, however, all these values are arbitrary cutoff. So it's you to decide what you want to go with, right? So people may say, okay, I want to go fold change higher than 2, maybe 3, maybe 10. If you have very, very high number of genes, difference expressed, then you may want to go p-value less than 0.05. You may want to go less than 0.01, right? Q-value 0.01. Or if it's very low expressed, then a very less number of genes differential expressed, then you may want to go p-value higher. Main hindrance of obtaining multiple replicates is often the cost. Yeah, and usually, more sample size is better. But again, when you do more sample size, you are overburdening your research in financially, and so micro experiments are still expensive for some regular laboratories. So we want to increase the replicates, but also we don't want to overdo our research experiments, right? So for that, we need a design of experiment. So before we go to do any experiment, we have to design the experiment first, and we may involve the statistician at this point. We want to know the sample size, the power, and everything, right? What is design of experiment? It is the art of balancing considerations such as skills, cost, equipment, accuracy, and many others. It's the art of balancing. And yeah, so if you increase the more sample size, the power would be better, but also it will cost more, right? And so that's why the art of balancing design of experiment is very important, and often requires to estimate the sample size for microarray experiments. How many replicates is enough? That is a common question when you do the experiment. How many replicates, how many sample size you want to have? It depends on the goal of the experiment and the resources and the availability of the technology, right? So there are certain methods which are more accurate, some are not. So there are multiple factors that affect. How accurate the chips are, that's also an important factor. How often the hybridization fails, that's also another factor. And how small the difference you want to detect. The smaller the difference you want to detect, you want to have a more sample size, more chip. Less, if you want to get the, if you want to get the bigger differences, then you can have a small sample size. How small is the noise level in the system? Also, noise variability is a very, noise. So if there is a noisy system, then definitely you have to have a more sample size. Different systems have different noise levels, and only way to estimate the noise is to do 3 or 4 replicate hybrid edges. And so this is uh, something I got this information from online and from online source. So like, for epimetric system with the best analysis, usually 3 to 5 chips per group or conditions. For example, knockout versus wall type, 3 to 5 chips uh, gives a useful information. And if you are going for if you are settling for greater than 2 differences between the 2 groups, if you are going for larger differences, greater than twofold differences, then 3 samples per group is enough. But if you want to get the narrow folds differences, smaller folds differences, less than twofold, then definitely you have to go for a bigger sample size, more than five groups per sample to obtain sufficiently reliable enough estimates of variations among samples within the group. And all these suggestions is based on the assumption that there is no any outlying samples, right? There is no any outlying samples, so these are the based on that. And if you are going with more like permutation test, and then you need to have more sample size per group, more samples per group. And it's always safer to add more samples, 1 or 2 more, especially in clinical situations where outliers occur commonly. In animal experiments, where often one animal is a conditional in a condition appears very different than all the others. So this is basically, you need to understand 2 things, right? One is, what's your sample, what's your type. If it's a cell line, then your samples are pretty homogeneous, right? So in that case you may rely on this. But if you're using the animal model, for example human, we have very heterogeneous sample. There are a lot of variables, a lot of factors, right? So this may not be applicable when in a heterogeneous sample. There is a chances of outliers very high, especially in primates and human samples. So you need to be very careful. This is just may not be true in all the scenarios. And again, samples and power, they are the 2 faces of the coin. Power meaning that power is the ability of a test to correctly detect the true differences, right? That's the power. And sample size and power, they are related. Like higher the sample size, the higher the power would be. And sample size depends upon many factors. And one is how small the differences you want to measure, or how big the differences you want to measure. And also sample size depends upon what is the power you want to get from the study. It's usually it should be over 80%. Power of the test is the portions of the differential expressions that are likely to be detected by the experiment. Sample size calculations also depends on other factors, like it depends upon the effect size. Effect size is a ratio of mean difference divided by the variance. So standard deviation, square of standard deviation is variance, standard deviation. So if there is a higher standard deviation, meaning that there is a higher variable in data sets, that is meaning that there is a noisy, then you need to have a more sample size. But if you have a smaller standard deviation, meaning that a small variance, lower noise, then you can have a smaller sample size. And usually in the microarray, it is considered to be, the standard deviation is considered to be 0.7. That's fairly realistic for genes that are expressed at moderate to high levels. And this is based on log2 scales. The sample size calculation need to estimate the standard deviation of the gene measurements on the base 2 log scale. So on the base 2 log scale, standard deviations are assumed to be around 0.7. And if you really want to do sample size calculation, usually people do sample size calculation for grant application. And if you are writing a grant, and if you want to include the microarray experiment for profiling the gene expression, or for getting a different gene expression for a transcriptomic study, then you may want to do the sample size calculation. And I found a very important, very interesting online tool from MD Anderson, and you can go to this link and play with this number, play with this web portal, yeah. And there are multiple factors that affects the gene power sample size. It affects the sample size, the power, or sample size. They are affected by the number of genes that is used in the array. How many number of genes? More the number of genes, you have to have a more sample size to get the same power. And also the number of false positives you are allowing, you are accepting. If you are accepting more than one higher, then you can go with the smaller sample size to get the same power. And fold change differences. If you want to go with the higher than twofold, if you want to go for a more fold change, if you're allowing capturing bigger differences, then you can go with a smaller sample size, for the same power. And desired power, like again, desired power is 80%, the 0.8 is 80%. Standard deviation also is affected by that. So if you are desiring higher power, then you have to increase the sample size. Sample size will increase if the desired power is higher. And the standard deviation, so if the standard deviation of your data sets is higher, then you need more sample size. There are many other factors, and especially like I told earlier, in a human, in tumor study, in identifying the breast cancer versus normal study, I have seen more than 100 chips being used per sample per group. So to increase the power, people use 100 of samples per experimental conditions. And so this is just the same thing I've gone through. You can you can study by yourself by playing with this web portal. And assumptions is that each gene in each conditions are normal, they are in a log scale, normal distribution on a log scale, and the variance between the two conditions are the same. Variance is the same in the two experimental groups. And so whatever the results, sample size you get, this sample size is like, say if you get the sample size of 20, then 20 should be in one condition and 20 should be in the other conditions. So result of the computation, the sample size per groups will be given. Now let's move to the other two types of microarrays. Broadly, there are two types of microarrays. A glass CDNA microarray is from Elegant, and there's a high density oligonucleotide microarray. That is a chip-based microarray from Epimetrics. And let's discuss what are these. CDNA microarray from Elegant, in there's sports. The probes are printed on a glass slides, and the probes have a pre-fabricated complementary fragments, and those fragments are longer than 2 kilobase per long. Whereas oligonucleotide microarray, they contains the spores, they contains the probes. Probes has around 25 to 75 base per long oligos. These complementary DNA or these probes, these complementary DNAs or oligos, those are to capture the respective genes, right? Each spores are designed to capture the respective gene. And these oligos being shorter, they have more specificity. Shorter probes, definitely within these probes, 25 to 70, shorter probes will have more specificity. They are more specific in hybridization. They bind more quickly, right? They bind more rapidly. And longer oligos, there are certain longer oligos, they are more sensitive to binding. Obviously, the longer sequences are more sensitive, more accurate, right? And shorter sequences are faster in binding, but they may not be that sensitive. That may not be that accurate. So oligos differences is this is based on cDNA is based on the longer cDNA sequences of 2 kilobase pair, and oligonucleotides are shorter oligos. And another difference is like this cDNA is based on the glass slides. This oligonucleotides is on a silicon wafer, and it's being fabricated, it's being made using photolithography methods. The advantages of cDNA microarray. What are the advantages of the cDNA microarray? These are relatively affordable with a lower cost. Hybridization doesn't need specialized equipment. Obviously, we need the glass slides and we need the longer cDNA sequences of 2 kilobase pair to target the gene, right? So it's lower cost, affordable, and hybridization doesn't need specialized equipment. It is not based on photolithography at all. And the data capture can be carried out using equipments that is very often already available in a laboratory, flexible to design as necessitated by the scientific goal of the experiment, right? And it's being a glass slide based method, it's very easy, and it uses the longer computer DNA sequences to target the genes. So that's why it has more sensitivity, increased detection sensitivity due to longer target sequences. But it has a disadvantage too. Disadvantage is its intensive labor, human labor requirements for synthesizing, purifying, storing DNA solutions before microarray fabrication. You have to prepare a longer DNA fragments, right? So it's more human intensive, more laborious. And there's a high chance of cross hybridization. You are using those in the glass slides and you are using a longer sequences, but the cross hybridization is very common. It is not based on photolithography, right? That's the one thing. The sequence there is a lot of homogeneous sequences. The homogeneous sequences have a very high identity, right? Homogeneous sequences may have 100% identity, but we went through like there may be a homogeneous sequences with very low identity, insignificant identity, but there are mostly in general, we say homogeneous sequences have a very good identity. So sequence homogeneous between clones representing different closely related numbers of the same gene family may result in a failure to specifically detect individual genes, and instead may hybridize to sports designed to detect transcripts from a different genes. So cross hybridization is possible in this kind of study. The next is the oligonucleotide array. It's on in situ on chip. It has a high speed with reducing the likelihood of contamination mix-up. Why high speed with reducing the likelihood of contamination? Because we are designing a very short sequences, oligos. So it is a very short, and the contamination is not, it reduces the contamination and mix-up. And we'll learn more about this, why it reduces the contamination and mix-up too, in the coming slides. So sporting of DNA onto the chip requires only that the DNA sequence of interest be known. Therefore, no time is spent in handling the cDNA resources. It is a high specificity and reproducibility. Each sequence is designed to be perfectly complementary to a target sequence, gene sequence. At the same time, an additional partner sequences is designed that is identical except for a second base mismatch at its center. Sequence mismatch strategy, along with the use of multiple sequences for each gene, increases specificity and helps to identify and minimize the effects of non-specific hybridization and background signaling. So this thing, we will be covering this in the coming slides. The next few slides after this will make this thing more meaningful. And it has high specificity because it has smaller sequences, small oligos, right? When you have a smaller sequences, it can bind very easily. It doesn't have like longer sequences will have a lower specificity. And also, it also has a perfect match sequence and mismatch sequences. Mismatch sequences is mainly to control the cross hybridization or to minimize the cross hybridization background effect. We'll discuss this in other slides. The disadvantages of array of these oligonucleotides is like uh, in situ oligonucleotide format, array format tends to have expensive specialized equipments. Obviously, we need to have a silicon wafer and photolithography method. It's a complicated chemical process, electrochemical process. Uh, so to carry out the hybridization, staining of labels, washing, and continuous process is a very complex here. And although short sequences used on the on the array confer high specificity, there may be decreased sensitivity binding compared with glass CDNA microarray. Obviously, the difference is CDNA, glass-based microarray has a longer CDNA, so sensitivity is higher there, whereas oligonucleotides microarray has a lower sensitivity, it has higher specificity. But to compensate the lower sensitivity in the oligonucleotide microarray, what it does is it uses the multiple probes. It uses the multiple probes to capture the same gene. So this will compensate the lower sensitivity. That is one of the defects of oligonucleotide. We'll be discussing this oligonucleotide in more detail. Now, we move from cDNA versus oligonucleotide microarray to this epimatrix gene chip. So epimatrix gene chip is a oligonucleotide microarray. I told you that epimatrix company no longer exists. It is now under Thermo Fisher Scientific. It was procured in 2016 in around $1.3 billion. But still, only Epimatrix is the number one in the business. Still, like this is the figure that's from the NCBIGU database deposits. As of 2007, it is a pretty old figure here. But still, and this figure shows that in compared to multiple different platforms, this has more than 80% whole market whole. But still, Epimatrix, Thermo Fisher Epimatrix is still the number one in the business. It's more than 80% market is held by this company. And why it dominates the market? The number of reasons. Because it uses high density of their chips. So the chips are designed to capture high number of genes, and genes are to capture each gene there are thousands, millions of probes. And right, and it has a sizable number of patents. That's the other factor, and another is the first to market presence. So when this is the first in the market, then it definitely has many advantages, and many research and commercial partnerships. So many hospitals, many research centers still use Epimatrix gene chips to study the gene expression analysis. For the transcriptomic analysis, it uses that. Each gene is represented in. I'll focus more on Epimatrix gene chips now because it's widely used. That's why we have to focus on Epimatrix more, right? So Epimatrix gene chip, each gene is represented by multiple short 25 base per oligo probe synthesized in situ on a silicon base. Microarray mRNA that has been extracted from the biological samples can be labeled, dyed, and hybridized on to a gene chip in a manner similar to that described for cDNA microarray. We already have looked. We have already looked at how the microarray works, right? In the microarray, in the two-color microarray, we discussed that. We showed that two samples are labeled with two different dyes are injected into one microchip in one microarray, right? So that is two-colored microarray. But this Epimatrix is a one-colored microarray. There is only one color, so only one sample can be hybridized in each chip, rather than the two in the case of cDNA microarrays. So here only one sample is injected, one sample is inserted, used for a chip. And there's a multiple probes to capture a gene. So per gene is multiple probes per gene. A probe set, I will go to this slide and talk about this. So this slide will show you the probe set. So this is the probe sets, right? This is a, if you look at this, this is a gene sequences. This is a gene sequence, and gene sequence can be pretty long. So it's given by dot, dot, dot in both ends. And you can design a oligo, short 25 base pair oligo. So this is one oligo, just piece of this, right? And this, and there is a, this is a perfect match sequence. And there is a mismatch sequence also is designed. The perfect, the mismatch sequence is nothing, but it's the same thing. Only at the middle, one is one base pair, there's a complement to that base pair. The G, complement of G is C, right? So only one base pair is changed to create the mismatch sequences. And this is the probe sets. This one probe set is used as a to capture one gene, this gene, one gene on at the top. And there are 11 probe pairs. So these probes comes in a pairs. So one is for the perfect match, another is for the mismatch sequences. This is perfect match, this is perfect mismatch. And there are altogether 11 probes, so 11 probe pairs, and these are the probe cells. One, so in a probe pair, there will be two probe cells, one for the perfect match, other for the mismatch. And there are altogether 11 probe pairs, right? This is for this segment of this gene, this may be for the other segment of the gene, this may be other segment of the gene, so altogether there are this. So do you understand this? So let me read this. A probe set is used to measure mRNA levels of a single gene. Each probe set consists of multiple probe cells. Probe set consists of multiple probe cells, and each probe cell contains a millions copies of one oligos. Right? This probe cells comes in a probe pairs, and each probe cell contains a millions of copies of oligos. This contains a millions of copies of oligos. Nucleotides. Each oligo is intended to be 25 nucleotide in length. Each probe pairs contains a perfect match PM. When you say PM here, it's a perfect match probe cells and a mismatch MM probe cells. A PM perfect match oligos perfectly matches part of the gene sequences, whereas MM, the mismatch, whereas mismatch oligos is identical to the PM perfect match oligos, except that the middle nucleotide, 13th position of the 25 base pair, is intentionally replaced by its complementary nucleotide. And there are 1 to 5 million probes per chip. How many probes? There are 1 to 500 million probes. So there are a lot of probes. In a tiny chip, there are so many probes, millions of probes. And within the probes, there are millions of copies of oligos, right? Now, this gives you a more better idea. After this, you get this. So this is a gene sequences, right? This is a probe pair. This is a probe set, and these are the probe pairs. One is perfect match, mismatch, and this is getting the sequence from this part of the gene, this is from this part of the gene, and so forth. This is for this part of the gene, this is for this part of the gene, you know. So different probe pairs represent different part of the same gene, and you don't want to select the gene sequence from anywhere of the gene, right? You want to select the place where there's a good chance of hybridization. There may be area where there is a repeat dimer, trimers, repeats of nucleotides, which poor gene sections where hybridization characteristics is very poor. You will not want to select those areas. You want to select those areas where the hybridization is very good, would be very good, right? So you can design the probes to capture the gene. Now, this is the pictures. I got it from the FU Matrix website, and this is showing you different probe cell with different oligos. So you can see millions of DNA strand built up in each location. So this is one cell, millions of copies of oligos are there, and oligos are 25 base pair long, right? This is strand here, you can see that. And the actual size of the chip is not that big, right? We're not talking about millions of probes, oligos. Its size is 1.28 centimeter by 1.28 centimeter. So it is, maybe the size of our thumb. Gene chip array. So this figure is showing you the probes, oligos, probes contains the oligos to capture the gene, right? And we inject the fragments which are labeled with fluorescence dye. So those would hybridize onto that probes, and you can see, so the binding, the fluorescence leveled sequence are bind to the oligos there. And upon shining a laser light at gene chip, that causes a DNA fragments that hybridize to grow. So you can see if there is a better hybridization, then the glow would be very strong. If there is a no hybridization, the glow would be very low or none. Overlapping probes within probe set. So this is about the same thing I'm repeating here. The gene, and you are designing a probe. So this is there will be 11 probe pairs to capture a gene, uh, and so there is a 11 times 2. There is a 11 pairs. There's 22 signals here for each gene expression. This is one. First is for perfect match. Second is for mismatch, and you can design the overlapping probes. Like this section is overlapping, but again, perfect match is perfectly copied from the actual gene sequence, whereas mismatch is only the middle position change. And you can design, and then after you excite the light and you normalize and everything, uh, you get the scores here: perfect match, mismatch. So you need to find a way how to summarize the scores, right? The way to summarize these 22 scores to produce one score to indicate gene expressions, there are multiple methods like mass4, mass5, there's a RMA, there's a plier, D-chip, there are many other methods. And usually the image processing is you get the gene expression, you get the image file, which is the raw image file, first thing, and then you need to do the background correction, normalization, and you get the real picture, real signal. So a cell file is the raw output in FE matrix. It's based on the intensity, right, for each probes. And one other thing you need to understand is, like, there's a different sports, different probes arranged in an array, in a FE matrix, in an array, in a microarray. So you need to understand which sports contain which genes, which sports is targeted to which gene, right? That is very important to know. And to know that, you need to know the annotation, how the sports are designed to capture which gene corresponds to which gene, which sport corresponds to which gene. You need to understand that, and for that you need to have you need to download, you need to have a annotation file. Each epimatrix array has a corresponding CDF file. This epimatrix CDF file links probes to gene. This is kind of a annotation file. And when you do this, we will download one of the required annotation file in our lab part and proceed further. Okay, and after you, usually in the epimatrix, the raw file would be the cell file, but after your background correction, after normalization, you get the signal file, which is normalized scores per gene that represents the gene expression. Those are called the chip files, CSV chip files. Raw input is cell, the correctly normalized signal files are chip files, CSV. And just like in the while you do the qPCR analysis, you use beta actin or GAPD, right? GAPD, some kind of controls. In the array also, there are different types of controls like RNA quality control. There's a sample preparation controls, hybridization controls, and there's other controls may be present depending on the array types. And you can recognize control probe sets by their IDs. So these you have to go through their user manuals, their manuals of each chip to understand what are the controls they used and how to detect them. Now, then accessing the quality of the data. Okay, once you do the normalization and you do transformation normalization, you want to check the signal intensity per chip, right? You want to understand the quality of the data. And you usually you do different kinds of plots like box plot, histogram, MA plots to see whether genes are, whether the distribution of genes are normally distributed, whether they are in the each chips has an equal level of distribution or not. You want to understand that. To that you make these kinds of plots. You want to know the signal, you want to know the noise level on the data sets. So these plots, this gives you a visual understanding of the visual picture of the data. Which software do you do to normalize? Usually you use bioconductors. There's a MAS5, there is a plier, D-chip, RMA, GCRMA, BSN, GLE, and so forth. In Epimetrics microarray, people use RMA. The MAS5, this MAS meaning that microarray suite, MAS5 is a version, 5 is a version. And there is a probe arithmetic intensity error estimation. There are these three methods, but among these, robust microarray average is a very widely used, very accepted method. So I prefer to use RMA method, and we will discuss about these methods. So RMA is it normalizes PM perfect match values only. So epimetric, you see the perfect match and also the mismatch, two different signals are collected, but it only uses PM, perfect match. It doesn't care about the mismatch spots at all. And what it does is, it first is from the cell file, it understands, it gets the PM intensities, meaning that it gets the PM value, perfect match values. Here it's from cell files, it detects the PM values. Then it does the background correction, background PM corrections, so corrections for spatial variation. Then it does the log transformation, log to transformation of those background corrected PM intensities, perfect match intensities. Then it does the quantile normalization of those PM intensities. So this will help to correct the variation between arrays and also equalize the distribution of different arrays. And also it does probe normalization, and this allows for correction for variation within probe sets. And the probes behaves the same way for on all the array. So what's the advantage in this multi-array, this robust multi-array approach is it not only corrects within its probes, but it corrects is done across different arrays. It does correction across different arrays too. Another is a MAS5. It is developed by Epimetrics in 2002. And this is the Epimetrics frequently upgraded this method. Initially, it used MAS4, and how it worked was it used to calculate the weighted average of the probe pair differences, so PM minus MM for each probe pairs representing a gene. So the perfect match minus the mismatch. So it uses the mismatch. It minuses it. It detects the PM intensities, this background noises, and then it takes the weighted average. And the improvement of this version, the higher version, MAS5 to this 4, is it basically did the log scale transformation before taking the average. The differences between MAS5 and RMA are like we already said, MAS5 uses MAS5 normalizes only array independently, each array independently. It doesn't do the like MR robust multi-array approach. It uses not only the within the probe normalization, but it does the array-based normalization to across array normalization, whereas mass5 only uses normalizes only one array at a time. Mass5 uses data from mismatch probes to calculate the robust average based on subtracting mismatch probes from values from match probe values. What it's saying is that mass5 uses the mismatch scores too, right? Mismatch intensities, whereas RMA method didn't use that. RMA method doesn't use the mismatch probes because their intensity are often higher than the match probes, making them unreliable to as indicated of non-specific binding. And another thing is RMA values are in log2 units, and RMA is more often used nowadays than mass5. Methods of analyzing microdata. There are multiple methods of analyzing microdata. One is using the Epimetrics software. If you are using the Epimetrics gene chip, then I would like to use the Epimetrics software itself. It is very easy, user-friendly, and it is designed for a bench scientist. You don't need to be a bioinformatician doing command line, scripting. It's a very easy to use. So I prefer to use Epimetrics software if you're using Epimetrics engine. Next is R packages. So you have a lot of flexibility, there are a lot of bioconductors available to do the different types of gene microdata analysis. Another simple approach is web-based GO2R tool at NCBI, and this is NCBI tool. And if you are doing the secondary data analysis, if you are analyzing the genes, the study that has already been published on in GEO NCBI, then you can use this GO2R tool to do secondary analysis. Next is there are lots of commercial tools out there, like there's a Pertec, Genomic Suites, Array Studio, GeneSpring Suites, and many others, but those are very costly, very expensive tools. Biometrics tools are very expensive, and if you can do it using the open source tools like shown in top 3, then you don't want to pay money like it. Let's move this Epimetrics software. Epimetrics GeneShip platforms is most popular and has remained as a gold standard for robust comprehensive expression analysis. Now it's called Thermo Fisher Scientific platform. Earlier, two tools were required for TAG 3. This is the earlier version, TAG 3. This represents transatomic analysis console 3.0, but now they have a 4.0, and usually in TAG 3.0 they had two different software. One was expression console, another was transcriptome analysis console. Now they have incorporated this into one. So there is no two tools, there is only one software called transatomic transcriptome analysis console TAG 4.0. Only one. And if you want to learn more about this, there is a YouTube link, and then you can get a lot of ideas, a lot of concepts by going through this. And there are many ways to normalize data, but RMA is the one which people usually pick. And input is a cell file, because the image file that generates by Epimetrics microarray is a cell file. Then the chip is after normalization, after background correction, after transformation and normalization, you get the signal file, that is chip files. Now this is the workflow. This is based on TAG 3.0. TAG 3.0. So TAG 3.0 had the expression console and the TAG software, but now this entire thing is considered as a TAG 4.0 TAG software. And first is import cell file, and does perform gene level normalization and signal summarization. Then identify and remove the outliers, and then redo the same thing and select the chip files, that is import the chip files, that is signal files now after clean files. Then you do difference in expression analysis. So you do alignment, you do mapping, and then you do difference expression analysis, you do pathway enrichment, pathway analysis, and you visualize it. Okay. Uh, uh, so most of the things, since this is a commercial tools, they have their workflow, their manuals are well documented. So Take Software provides ability to normalize, perform data quality control, and summarize data from thousands of samples, tens of thousands of genes, hundreds of thousands of exomes, or thousands and hundreds of thousands of different transcripts isoforms using a standard desktop computer. Usually simply using our desktop Windows computer, you can study the isoform, you can study the gene expression, and you can study the interaction, microRNA interaction, you can study the pathways. So many things could be done using this tool. And RMA is the summarizing algorithms that you want to pick from here, and it provides a powerful visualization tools like PCA plots and signal histogram, scatter plots, box plots, and also Pearson correlation plots. So it's just by clicking you can get all of these plots. And this is a very good, but one defect here, right? So you may have a computer with on different platforms. You may have a Linux-based computer. You may have a MacBook, a Macintosh computer, but it only works on Windows PC. And I don't know why they didn't incorporate it. They didn't change their software, make their software adaptive to other platforms. It's only available for desktop only. That's the only drawback I found, and in fact I told one of their visiting scholar or visiting people, but they don't want to do that. They want to keep it simple for the desktop users. Take expression console software. Creates a chip file. So expression console, it creates a chip files, and it transfers it. It should be transferred to the Take software, but the transformation is so smooth that it doesn't. We don't have to do much work, but because of transformation need to be done, this has been avoided in the new version. So you can simply download Take version 4, and you don't have to deal with this. Input the chip cell file and get the final expression results, by eliminating the expression console. So with Take version 4.0, expression console is integrated into a single application. That's what I was trying to say. So Take 4.4 software developed by Bioinformatics enables you to go beyond simple identification of differential expression by providing visualizations of complex gene pathway networks, microRNA, and target gene interactions, alternative splicing events. So these are the things this Type 4 software has capability to do. And this software can also, good to visualize the biological pathways. You can visualize the biological pathways. It uses the wiki pathways. So it doesn't use any other pathways, but if you want to, it will show you which pathways was expressed, and then which pathways and how many genes from the study has been identified in that pathways. You can dig into that. You can understand the pathways. It will take you to wiki pathways page, and it will help you a lot. And also you can do the statistical analysis, like you can do difference in expression analysis. You can identify which genes are differentially expressed, or which exons are differentially expressed. Also, it will give you which genes has alternative splicing events, and even you can see the pictures of the alternative splicing events. Visualize gene models with exons and junction signals. Visualize fold chains of gene levels and mRNA, microRNA, link directly to publicly available public annotations. Take software, yeah, it like already I said you before, it can do scatter plots, volcano plots, hierarchical clustering with heat map plots. It can do chromosome view, so it can show you, which genes are expressed in a chromosome. It can visually show you that. It can show you which pathways are expressed, like wiki pathways, and then alternate splicing events can be seen, and also you can uh, map expression changes between mRNA and targeted genes. This is something which you like. This is after you. This is the flow, but here the picture is what you get the end product. So like after you do the get the results, after you have a successful run, you get all the columns of IDs, transcript IDs. So these are the probe IDs, and this is the signals for one sample log 2 signal. This is the average signal, and this is the average 2 signal for another samples. There's a pole change difference in a linear scale, and there is a, see, here it's done ANOVA. And if you are using 2 samples, then it will give you the t-test. If you are designing multiple samples, if there's a more than 2 samples groups in your design, then you get the ANOVA. Then yeah, then you get the FDR p-value. There's a Q value, and then gene symbol. Gene symbol. You may have multiple gene symbols here because the probes may be designed in such a way that it can capture multiple genes, too. So, and there's a description, and there you can click here: scatter plot, volcano plot, chronological summary, equipathways summary, and all those things. And this is the another one. This is the GO2 base GO2. Uh, this is NCBI's web-based portal. GO2R tool is there, and this is for the secondary data analysis. It's already is for the paper that's already been published and data already been uploaded to GEO. So you can find based on the gene accession number, GEO accession number, GSC, gene series number, you can identify such a gene, and then you can simply define the different levels here, and then you can do the data analysis. And what it does is, it is very easy to use. Very, it doesn't take that much time. Within a manner, minutes, you can get a lot of information that you can capture. And also, it generates the R scripts for you. And that R scripts you can download, you can copy and paste and use it locally to extract the data from GEO to R. Next is Bioconductor R. R is a free software, right? And also Bioconductor is one of the is also tools, is a free available tools that mostly based on R. Bioconductor is mostly based on R. It is used for high throughput genomic data analysis and microarray data. For microarray analysis, there are a lot of bioconductors available to do data analysis. So before I move to the lab part, the experimental part, and I think I provided you the 3 different approaches, like the 3 different approaches. So this the one is based on the Epimetric software, another is based on R packages, another is based on the web-based GU2R. So lab 1, lab 2, lab 3. So lab 1 is based on GU2R, right? It's very easy to use, so use this at first. Then go to this Epimetric software and then download it, install it in your Windows computer, right? Not in Mac. Mac doesn't have that. And then you can do that analysis. It's very easy to use. Whenever I get the projects on microarray related to Epimetrics, I don't use any other tools. I just use Epimetric software, which is very efficient, very easy to use, and it gives you the publishable results. And next is R packages. There's a lot to learn. This is a big topics. This is these things contains a lot of bioconductors. You can learn by exploring these packages on your own. Okay. First thing is lab GO2R. So this is a very easy to understand method. It's a very easy to understand method. The difference, the methods for analyzing microdata. So this method is web-based GeO2R tool at NCBI. There are many other methods which I've discussed, but this is number one. And I've given you something to read on that, but I'll show you here how to use it. Okay, you just open a web browser and then maybe type GeO2R. This should lead you to something. Yeah, it will give you. It will get you to this picture, this link here, NCBI GeO2R. Okay, so this is a NCBI GeO2R tool. So you need to select SSN number. So SSN number, you know. So I've selected this SSN number. This is the SSN number, GSE1397. Then set. So you see all the studies, all the microarrays that's related to this study are shown here. There are 28 samples here, and this study is about trisomy 21 and TS 13. It is about like this is about the TS 21 human trisomy associated with Down syndrome, and there's a process eupolite normal chromosome copy number samples. This is a study about this. So if you want to study more about this, then you can find this paper, find this project, and explore more. Okay, so you can study this about this paper, and now you need to define a group. Suppose if you want to study the difference between trisomy 21 versus eupolite normal chromosomes, then what you need to do, you need to define a group here. So I've already defined a group here. So maybe, maybe because I ran this before, that's why it already appeared for me. But if you want to say, okay, so if you want to give a lever and then just hit enter, it appeared lever here. But I don't need any lever, right? I just close it, okay. Okay, now I just have two groups. TS21, eupolite. Oh, sorry, I already selected this. It's already been done. Okay, I'll redo it. So what he's saying is select this access number and hit set, right? And then you get this list samples here. Now you define a group. You can define a group using TS21 group. Enter. You get this. The other group I want to define is EUPLOID. Eupolite group. So in that, I want to select all trisomy sample and eupolite sample. So I will just, so this is, so I've given you the list which to pick, right? So this one is TS21. This one is TS21. This one is 2015. I can hit shift, select shift, click 21. This is, and this is 23. 23, shift 24, 26, 27, 7, 83, 84, 85 is your deployed. Then again, you can select this 86, 87, 88 to TS. This is TS21 group. Then this is 8, uh, this one, and then 64, and this is diploid group. Then this is um, so altogether how many? There's a 7, 9. I need to have it equal 11. So this one is TS6, 8, and then this is TS21, and then 13, 14 is TS21. So there's 11 samples in TS21. In the euploid, there is this one, and this one goes to euploid. So altogether 11 samples. So there are many other samples, but for this study we can just take 11. Okay, so I defined the two groups. Then it's simple. Now what I need to do, I can just hit top 20 to 50. And it's starting to process it. It's starting to process it. Then, okay, now I can see all the information that I need to. So based on that, two groups, I get this probe ID number, and these are associated with genes here, gene symbols given here, right? Gene symbols, and this title, what it means. And then I'm getting the adjusted p-value, which is Q value, and I'm getting P value, I'm getting the T statistics, I'm getting the B statistics, I'm getting the log fold change, right? So based on log fold change, I know it's up-regulated or down-regulated. We can, if it's a positive number, then it's up-regulated. If it's negative number, it's down-regulated. And I know it's significant or not significant based on FDR level or not, right? And if I want to see the profile of each, like say this is a SOD, right? SOD1 is a sodium dismutase. It is a reactive oxygen species group, right? This is antioxidant. Okay, so this is, if you look at this, then I can say, oh, okay, TS21 have a higher expression than euploid. So this particular gene are highly expressed in this group than this euploid group. So you can get this, and this is something we can do using this profile plot. Just if you give the IDs, you get the same thing like you are getting here. And then you will get the distribution, like how our normalization was done, and how the arrays different samples look like in distribution. So it seems like pretty much normal, I would say. The median is pretty close to each other. There are 11 TS21 group, and there is a 11 euploid group. So you can see this thing here, and you can export view. You can do a lot of things here. And the option is like, what are the options? You have very limited options here. You just can select which method of false discovery you want to use. Like usually it's using Benjamin Hosberg FDR value, right? So if you want to move to the other Boniforni or any other methods, then you can pick here. Yeah, and this is NCBI generated submitters. So this is something which you can play with. So there's not much to choose from here option, but this will give you the R script. This will give you the R script. So if you copy this R script, see, if you copy this R script and then you can run it in a computer in a R, then you will get the same results. You can modify it, you can change it, you can do a lot of things from here, right? So it doesn't have to depend on limited capabilities given here. So what I tried was like, I just copied the same code here. It's version R, everything. So So this is, see, look, I copied all the codes here and I ran, and I was able to get some figures, some figures, figures. Maybe I'll clean this up. Okay, I'll clean my plots. I don't know why it's saying server error, but okay. Is it doing some? Oh, I don't know. I should not have installed the. I'll just run from here. I do have already installed BIOS. I have Geo2, I have Lima, and then I'm running whatever given there. So it gives me all the results. It will write down this. I'll get all the results here. Yeah, I don't want to write it down on my computer, but I want to show the box plot. Right? Maybe it's good to show you the box plot. So this is based on like ones in this list. These ones and zeros are the two groups, but x are the remaining samples which we did not want to analyze. Right? There were 24 samples, but we just used 22 or like 20 samples around that. We just wanted to use 22, right? So that's why x meaning that we did not want those samples. Okay. So see, I got the same results in my my screen here. Okay, so this is about this. So it is very easy. You can try this at home. Okay, so now I moved to my Windows laptop. So this is for the Epimetrics. Okay, I want to show you how to use the Epimetrics software. Okay, so I've given you this instruction, this some kind of a note how to do this Epimetrics software. So this is about the Tech 3 software, but the fundamental difference is known between the Tech 4.0 versus 3.0. So there is nothing much difference except the two tools has been combined to one. So you can go through this, everything. And then if you want to learn more about this, then there is a software link, and then all what I've explained in the class, it's all in here, right? I've made these informations to the slide. So everything, most of the things are here. Okay, so the instructions, the lab part, the top earlier parts are more for you to study. The later part is the lab part, labs on microarray analysis, lab and microarray analysis. So first is download the software. Tech software need to be downloaded. The tech software downloading is pretty easy, but you need to have a login. You need to use you have to have an email ID, a Hawaii email ID or something like that. And then you can download the data sets, but I've already provided you the data sets from one particular chip. The chip is STA 2.0 array. So I provided you the cell files from this array, and you can download from Lima. And you can open Tech 4.0. Since I've already downloaded, I've already installed, I can do that. So if you look at my screen, then I do have Tech here, Tech software here. Just let me minimize this. Okay, I'll just close this, and it's in my desktop. And then, okay, I'm opening my Tech 4.0, Tech 4.0. Now, I have already downloaded all the cell files. It's in one of the folder. So now I'm ready to import it, right? So import file is here. So I need to import files. So I can import either self files or chip files. You know the difference, right? Self files are the raw files, which which are not normalized, not background corrected, not transmitted, not normalized. But chip files are. Chip files are after normalization. So you can either input chip file or shell file. So since we haven't done anything, I'll just input chip the shell file. So these are the shell files. So these shell files are uh, are this. I've kept this in my desktop folder, and it's inside the desktop folder. I've made like folder like uh, like transitomic in my desktop is a transitomic essay analysis suite 4.0, and I made multiple folders. I keep the cell file here. So these are the cell files. So it's a liver cell file. Liver is one type of samples, and other is the muscles. So I will copy all of them, import all of them, open. Now I'm ready to open it. So now it's importing it, and are you guys seeing this? Okay. So yeah, and there are so many things to play with here. So analysis name, I can give my own analysis name, and maybe analysis class projects or something like that. And then where the results I want to keep. I want to keep in my document, or I want to keep it inside the. I can browse it. Where do I want to keep it? I can keep it in different folders that I want. Then next is here. So you want the gene plus exome level difference, or you just want the expression for gene only, or you want to see the expression between genes and also expression between exomes. So if this is important. If you are going with the alternative isoforms, then you need to choose gene plus exomes. If you're just you don't care about the alternative splicing events, then just go with gene, right? The expression, gene only, gene level only, not the exome level. And then this is like a robust multi-array transformation. You can go with the default one, the robust RMA method, robust multi-array method. And then here, the conditions. So we are going for two group comparisons. So it is not, you can do repeated measure ANOVA, you can do batch effect, you can do normalized group, you can do random factors, real, there's so many other choices, but our study design is pretty simple. It's two group, liver and muscles. So it just keep comparison group. So you can change the name here if you like, right? You can add a column here, like you may have multiple other groups, right? So but here, since it's very simple, I just keep it in a simple way: liver versus muscle. Okay, now I need to select everything, so all the things selected, so it's liver versus muscle. I want to do comparison. I want to see which genes are differentially expressed. And if you want to set up different algorithms parameters, then you can click here. So you can have what's the criteria for calling default setting, criteria for selecting significantly different. You want to go with false is greater than 2, less than 2, less than -2. You can have a p-value 0.05. You may want to select FDR or not FDR. So here, if I let it be, I don't want to go with FDR, right? So I just put FDR false. I don't want to select the best based on the FDR, but still I want to uh, call FDR here. And then, so it is using a Lima Eways method. And then, so yeah, so you can look at these different criteria here. Just keep this as default. And uh, comparison wizard setup, we don't need to do anything much because our study is pretty simple, easy to understand, easy design, right? Just hit run analysis. Then it will take some 1 or 2 minutes. 1 or 2 minutes. Let's see how long it takes. So it's doing, it's already took more than 1 minute now. It's doing some, it's working on liver, muscles, beta. Okay, it's working on the, okay, it's muscle. It's running a probe summarization, okay. Running probe set summarization still. Before doing this, I forgot to tell you one thing. I forgot to tell you about downloading the annotation file, because the file will not be uploaded, imported, unless you have that annotation file, annotation, the CDF file there. So let me finish this, then I will show you how you can do that. Now it's creating prop set ID list, creating prop IDs. Okay. We're running expression analysis now. It's close to be done. So it's using Lima, writing results, reading annotations. So it's reading annotations. So annotation is very important. Now it's done. See, it's done. It didn't took that much time, right? It took me 2 minutes max. But I forgot to tell you one thing. Before I move to this, I'll just tell you one thing here. So this is, I can close this. Now this is done. So when you import it, you need to upload, you need to have that, that ready, that, okay, I'll show you that meaning, what that means. So you have to follow this. So you open Take Software, then you have to go to Preference, download library files if needed. I already have downloaded. So for me, that library file was already in there. So I did not need to do that, right? So what is a library file? It depends upon the chip. The chip, like if you're using different chip, Chromium, different version, then you just need to install it once. Chip meaning that your chip has a different sports targeting different genes, right? But you need to have an annotation which sports represents which gene. If you don't have that information, then then all the information is lost. You cannot do anything. There's no meaning to that, right? So you have to know which sport represents to which gene, which probe sets is designed to capture which gene. You need to know that, and this file has that. So it varies from chip to chip. So for example, if you're using STA 2.0 array, then we need to find this library files for this one. So how to do that? First thing is you need to create the account here. You need to create the account using this. You need to create the account, Control V. Why Control V? No, it didn't work. FA matrix. .com/site. Let's log in. Yeah. So you have to create a login account here, okay? And once you log into this account, once you log into the account, so what you need to do is you have to go to preferences, and you have this your username, your password. The password, once you log in using the link that I provided you in the note, then you can download the library files. So already my password is pretty active, so that's why nothing has, I was able to get this information here. So usually if you are using a human D, Chromium D chip in human is pretty common. So I've already installed it here. It's already installed it here. And for this particular analysis, I was using STA2 chip, right? STA chip, STA chip. It's an STA chip. So I use this, click and then download. So it's already installed here, so I don't need to do it. So if beside this, if I want to install, if I'm going to if I have a Chromium mouse chip, I'm using mouse chip, then probably I need to have installed this. Click here and then download it. So I have used this before, so it's already already done. You know, maybe I'll just for example, I'll just show you here. Then click download. So it will start downloading. It will start downloading. So then you can upload your chip cell files. Then once you finish doing, after you upload, once you did that, then the end result is here, already done. We already finished that. Okay. Maybe I just want to cancel it. Yeah. Okay. So this is the result that I got earlier, right? So this is the result I got earlier. So what the result I got? Let's look at this. The results I got here is the sequence sample QC quality overview. This is QC overview. I can see the PCA plot. The two samples were clustered very separately apart, right? You can see those things. QC very different. Yeah, you can see the QC, and then you can see level controls, different controls, negative controls, box plot. Usually box plot seems to be pretty normal distributed, median's pretty, uniform, and it's in the same range. This is for QC. Then if you want to summarize views, then you can see, okay, these are all the things what you have done. So array type used was STA. When was the experiment, the analysis done, the date and time is given here, and then all the things you chose. So analysis type was expression gene. The summarization method was the gene level, SST, RMA, robust multi-array method, and then all the things. And the annotation file used was this, right? The mapping file, and then conditions. There were two conditions: liver and mouse. And for fold change, we considered greater than or less than greater than 2 or less than -2. P-value 0.05. Method was EVS method was used. So you get many info, and then you see that how many genes were upregulated, how many genes downregulated. There were 4 samples in each group, right? There were 5,000, more than 5,000 upregulated, 4,000 downregulated. So 10,000 genes differentially expressed, which is a very big number. It's a very big number. Okay, then you can go to gene list and Venn diagram. But when you have multiple results, and you can create a Venn diagram. Where here, since only one comparison, there is no Venn diagram here. Gene view. Now this is very important. Gene view. Did I go through summarizing table? Yeah. So this is the summary table. So this summary table is just 4 samples per group, filter criteria twofold, and p-value less than 0.5. And there were so many genes differences expressed. 15% of the genes were differentially expressed. That's a lot. And around 5,000 genes were upregulated. Around more than 4,000 genes were downregulated. And this is showing how many red is non-coding genes. This is non-coding and this is coding. So it give you this way too, coding, non-coding, and then yeah. So it's giving you just different uh, cases, coding and non-coding. Now moving to this gene table, and okay, let's understand this. So this is the IDs, the probe sets IDs. This is the liver, average liver samples, log to normalized, transformed. This is log to transform muscle average, and this is the pole chains. And this is the p-value. This is the FDR p-value. This is the gene symbol and description and then groups. So gene description meaning that what this is a TTN gene, and there is different kinds of genes, right? So and there you can groups coding, and also there's non-coding, should be non-coding and coding, right? Okay, and then if you want more add more columns, then you can click here, and then there are so many. Yeah, customize annotation. You can do customization. You can choose different, but let's go with the default only. These are the default, but you can add more columns, more annotations to these genes or this process. Okay, now this is too much, right? This is 10,000 gene. So what you can do is you can add a filter, right? You can add a filter here. You can add a filter. How to add a filter here? Filter, manage filter. You can add a column. You can add a filter here. You can make the FDR filter here, okay. But you did not choose any. We did not choose any. Manage, you can clear the filter, manage the filter. You can do a lot of things. So add a column filter here, FDR as a state. Maybe we're gonna go with 0.0. Since we have a very high number of genes, let's go with 0.001. Let's see what it goes. So it changed the number counts from 10,000 something to 10,001. Still, it's a very big number. And then you can play with this. You can go with maybe 10-fold. Just for example, choosing 10-fold. Yeah. So now districtly we are capturing more differences, bigger differences. We end up getting 2,000 genes, and based on FDR p-value less than 0.01 and pole genes greater than 10. And you can see the things changed here. This is scatter plot, and these are up-regulated genes. The red ones, the green ones are down-regulated genes, and these are very interactive plots. Then you can see the volcano plot. This is up-regulated, this is down-regulated. The one meaning full change, one meaning no change, right? One is, so these are up-regulated, and this is, you can do the hierarchical clustering, so you can get higher clustering. It says, please wait. Okay, I don't want to wait, maybe. Okay, now I got the hierarchical clustering based on 2 samples. This is one is muscle and liver. So it's so good interactive heat map plots. So you can easily see the differential expressions. So these are down-regulated in the here it's, let me see. So here red means high regulated. Red are up, and blue meaning low, right? 20, 10, 2. So these are low expressed in the liver. So all these are low expressed in the muscle. So if you consider these genes, then these are upregulated in muscle, and these are downregulated in muscle. And you can get the chromosome summary. So many genes up and down related per chromosomes. There's a human sample, so there's a 22 plus XY chromosomes. You can see that, and you can get the wiki pathways. So you can get the wiki pathways. It will take some time, so I will not focus on this. Hopefully it will be done faster. I don't know, because we just selected the, let's select on the significant scenes. Let's see, I don't know how things works. Okay, then if you want to export this, right? So then here's the export column. So what do you want to export? You want to export all the only current tables, or you want to only the current tables, or you want to export all the rows and columns. So I usually what I do is I export all rows and columns. It not only includes significant, but it also includes the insignificant express genes, differentially expressed genes. So I'll just export all, and then you can select where you want to select. You want to select the place, and then maybe give a name like results.txt, and then hit save, and then you can open that file as an Excel file, and then you can do the further analysis. Okay, so this is all about it. You can do self-exploration. Maybe I missed something. Many things can be missed because this is we can do a lot of things with this simple tool. Okay, so you can carry on with this.